This is an expert panel session on building a rapid, targeted, and responsive infectious disease system discussion. My name is Aaron Pomerantz. I'm an associate director on the global segment marketing team here at Oxford Nanopore. Now, many of you were probably in the session earlier. You may want to use your headphones. There may be some ambient noise. So just a quick reminder that you may want to use those, and if you have any issues, please just raise your hand and one of the staff can come by to assist. In the session, we'll have 4 distinguished speakers who will each in turn give about a 5-minute introduction to their area of work. This will be followed by a longer panel discussion where we'll aim to explore the future challenges and potential opportunities of fully realizing the power of genomic sequencing to improve human health. We'll be taking questions from the panel, from the floor, and via the app. If you're using the app, please remember to use the thumbs up icon and vote for your favorite question. I encourage you all to consider questions for the whole panel. However, if you do have a question for a specific speaker, please do let us know who to address with your question. Now, we ready? Excellent. So with that, I am now delighted to introduce our first panel member, Dr. Inez Hassan. Inez is a pharmaceutical and global health advisor with over 15 years of experience in strategic consulting in commercial and pharmaceutical strategy and global health policy. She is the director of the Global Health Security Consortium, which helps leaders prepare for health security challenges and with life sciences innovation. She has an engineering degree in biochemical engineering from the University College London and a postgraduate diploma in global health policy from the University of Edinburgh. Please give a warm welcome. Welcome, Inez.
Thank you so much, Aaron. And I thank you so much for inviting me to join this panel. And I'm absolutely delighted that I'm first, because the first thing I want to say is I'm not an expert in infectious diseases. So I'm very lucky to be among these panel experts who really are. But I come to this with a slightly different angle. So I'm based at the Tony Blair Institute for Global Change, and I work very closely with the Ellison Institute for Technology. And so here, what we do is we work very closely with global world leaders all around the world, and we try and keep certain areas high in their agenda. So for me, very much trying to keep pandemic preparedness, global health high on the agenda, and the further and further away we get from the pandemic, the harder it becomes. So that's really my goal. So helping governments think about global health priorities, the policies they need, the regulations they need in place to kind of enable the adoption of all of this great sort of innovative technology and to enable disease surveillance in peacetime as well as during an emergency. And at the Ellison Institute for Technology, which is headed up by Sir John Bell, who we work very closely with, we're focused on developing and deploying a lot of this innovation, and we're doing proof of concept pilots in countries, which I can tell you more about later on. So the great news is that we've got lots of really, really great innovation and technology that's becoming more and more portable, user-friendly, requiring less specialist expertise, and becoming more and more cost effective. I won't say we're there when it comes to costings, but in the world of global health, but it's getting better and better. But the trick is, how do we get a lot of this innovation and technology into the hands of those who really need it in the resource limited settings? And how do we invest in the much needed, and I guess not as exciting, but infrastructure to enable kind of the use of all of this kind of great technology as well, to ensure that we can have this targeted and responsive infectious disease system. So at the Tony Blair Institute and the Ellison Institute for Technology, we're focused on an approach we're calling the always-on approach to surveillance. And this approach is trying to see how we can embed surveillance into routine care so that we can pivot and quickly scale in the instance of an infectious disease outbreak, which could turn into a health emergency like another pandemic. And we're also trying to see how we can link the data as much as possible, which is very tricky, so that you can use it for individual care, population health, and clinical research at the same time. I don't have the solution to this, and it's very, very tricky, lots of political issues to get this right, but this is what we're advocating for. And also, the good thing about this is that if we can expand the use of surveillance to more than just infectious diseases or emergency use, it becomes more and more cost effective and more palatable for leaders or those who are making the investments in these particular areas. We're also trying to shift where this so that we focus on more multi-disease testing to make better uses of the resources that we have in place as well. And this will also require better data standardization, interoperability, better digital infrastructure, like I mentioned earlier. And ideally, we want this sort of next generation sequencing so that we can identify unknown pathogens, drug-resistant bacteria, and also to support vaccine development as well in peacetime. And really important to this approach is to move away from siloed projects and more into sort of more collaborative approaches across academia, industry, public health institutions, and across countries as well. Again, very, very tricky to do, but this is really what we're trying to advocate for. And obviously also to have more sort of proactive programs that target geographic hotspots, especially where we don't have the right infrastructure right now, and to predict and target where we might see sort of future pathogens arising. And we are working with some governments. I can talk to you a little bit about a project we're doing with the government of Rwanda, where we're trying to test this particular approach. And so we're sequencing at the moment. We can't do everything, of course, in our pilot, but we're testing sequencing for a couple of different pathogens: influenza A and B, measles, Klebsiella, and Salmonella. And we're trying to basically connect this central district facility, or the Rwanda Biomedical Centre, for those of you who know it, with a couple of district level facilities. And the idea is just to show that you can actually do a lot of this work in the more rural settings and actually closer to where you are going to see some of these outbreaks. And also it'll be the first time that Rwanda will not necessarily have to rely on sort of a national reference lab to do a lot of this work. But that's not the only solution. I think overall there's lots of things you need. We need the tech innovation, and we've got a lot of the people in the room who are focused on that today, but critically, especially in resource limited settings, which I'm interested in, we need the right funding. We need cost effective products. They need to be truly scalable, and my litmus test is that you should be able to use it in a refugee camp type setting. So I know it's hard to do, but that's kind of my goal. We need more research and development in this area. I said earlier as well, we need the right data infrastructure and interoperability. We need real-time access to this data. We need right governance. We all know that the pandemic agreement is being negotiated, and we're not sure how successful that's going to be, but we need the right governance in place to be able to ensure that people do get access to the data and quickly. We need the right rapid response mechanism, so we need a plan in place, quickly deployment of financing, and also financing in peacetime as well. And investing in the digital infrastructure as well. We really do believe in boosting parts of the health system which will improve the life science and health industry in general, and use that to have better responses in emergency scenarios. Very importantly as well is to have a community involvement, so educating the right people, making sure that you have sustainable training. One of the big problems we have in global health is that some people come in, train, set things up, and then move on, and equipment gets mothballed, and that training goes nowhere. So ensuring these are sustainable is incredibly important. And the last thing I'll say is that supply chain and logistics is incredibly important. In Rwanda, for example, we've had problems months and months to try and get reagents that we need in order to do some lab work. So looking at the supply chain as well is also something that we're focused on as well. And thank you.
Thank you so much, Inez. That was a great way to kick things off. And come on in, we got plenty of seats right over here. Now, before I introduce our next speaker, just a quick reminder, please keep your questions coming in for our panel discussion, which again will take place after all of the panel presentations. It is now my pleasure to introduce our next panel member, Dr. Chuck Cooper. Chuck is EVP and CMO at bioMérieux. He is a graduate from Georgetown University School of Medicine and specialized in infectious diseases and epidemiology at the University of Maryland, Baltimore. He's been practicing medicine for over 25 years and still devotes 2 days a month to this work. He was previously Chief Medical Officer at Siemens Healthcare Diagnostics, and he also spent 8 years as the Head of Medical Affairs at Becton Dickinson Diagnostics and 13 years with the US FDA. Welcome, Chuck.
Thank you, Aaron. So I wanted to share some thoughts about sequencing and the management of infectious diseases. It's obviously very exciting what's happening with tuberculosis. Huge amount of progress in a very short amount of time, and it's really amazing to see what the WHO just announced a few months ago with regard to recommendations on the use of it. But what about sequencing, more broadly speaking, in other areas of infectious diseases? I think we're at a moment of transition in this space, which is really exciting. So if we think back to the past 10 years, um, essentially the questions that people have been asking is, can we do this? Can we use metagenomic sequencing for the management of infectious disease? And how do we do it? So if you look at the publications, they're mainly focused on things like methodology. There are clinical studies that are mainly observational in nature, trying to understand, is there possible value here? So you see case reports or case series or cohort studies. These are all observational in nature. And they're very important because they lay the groundwork for what's to come. But when you look at the performance of these observational clinical studies that have been done, you see it's all over the place. Some studies have really high performance, other studies have very low, poor performance, and everything in between. And why is that? Well, these are observational studies. There's inherent bias that you can't control or you don't even know about. Patient populations are different. Endpoints are different. And even when they're the same, they're measured differently. So you have a lot of variability that creates noise. So how can we look at all that data and all that noise and understand clinical value and clinical utility? Well, the answer is that we can't. We need different types of studies to actually understand clinical utility and clinical value. And so that's the transition we're undergoing right now. And so when you look in the literature in the last 1 to 2 years, you see different types of publications. So there was a really good publication in the Journal of Clinical Microbiology by David Gaston from Vanderbilt University, where he proposes a framework for how we can think about measuring and evaluating value, clinical value, in the use of metagenomic sequencing in the management of infectious diseases. He also makes the point that usually for clinical utility, we use outcome studies to understand clinical value and clinical utility, but there are precious few that have ever been done, if any, of high quality. But that's changing now. So if you look right now in the last 1 to 2 years, we are now starting to see studies that are really aimed not on the question of how do we do it or can we do it, but instead on the question of how can we prove value? How can we measure value in the management of patients with infectious diseases? And so you have a study in Taiwan and in China and one here in Europe that have just been either started or ongoing in the last 1 to 2 years, where they're doing randomized prospective controlled clinical trials. So they actually randomize patients to care with conventional microbiology methods informing the management, and other patients randomized to conventional microbiological methods plus metagenomic sequencing. And they're following these patients and actually measuring real patient outcomes: 30-day mortality, admission to the ICU, duration in the ICU, antibiotic utilization, real outcomes that actually will inform and help us understand the value of metagenomic sequencing in the management of patients with infectious diseases. And this is really a critical moment. And so there are a handful of these studies happening now, but it's going to accelerate, and it's going to be the direction that everything is going to be moving. And that kind of information will be critically important for us in terms of understanding what role this will play in the infectious disease space. It will open the door for us to think about how we can commercialize these kinds of diagnostics and how we can make them more generally available broadly for the care of patients. So I think this is an extremely exciting moment in time and really looking forward to what's to come in the next few years. And that's what I had to share. Thank you very much.
Thank you so much, Chuck. And please remember for everyone in the audience to keep your questions coming in using the app for our upcoming panel discussion. With that, our third speaker is Dr. Rahul Batra. Rahul is a U.S.-trained infectious disease physician with a particular interest in the development of novel and rapid diagnostics. As Deputy Director of Clinical Infection and Diagnostic Research at Guy's and St. Thomas Hospital, his research focuses on development of clinical sequencing diagnostics for frontline deployment, including pan-metagenomics. Welcome.
Thanks, Aaron. Thanks for having me and for inviting me. I just want to talk a little bit. We've had really nice introductions here, and I want to talk a little bit about how we came to sequencing. I've had the pleasure to work with Jonathan Edgeworth for more than 10 years, and in that space, we started together in collaborating on hospital-acquired infections. And around the 2005, 2006, Jonathan had found a highly infectious strain of MRSA in the ICU, and we set on trying to understand what the parameters of those that made that infection so bad. And we wrote a grant together to actually have that sequenced. It took about a year to have it sequenced and bioinformatically put together. And we got a lot of really important information about its infectivity, potential vaccine targets from this single sequence. And at that point, we kind of said to ourselves, that's great, but wouldn't have that information have been important if we could have had it on at any time during this outbreak, not a year or two later? So we kind of put that to the side, continued our work, and in 2017, the NIHR approached us to say, would you like to look at sequencing technology for infectious diseases? And we kind of revisited some of those conversations about what would that mean now? What does it actually mean to frontline therapy if we had this sequencing technology? And we were both fairly skeptical that we could make a new technology work, and especially a technology that was just coming out with Oxford Nanopore. But we thought, we'll give it a shot and see what happens. And I think both of us were big skeptics at the time. It only took a couple of samples. It only took a couple of real patient samples and patient time to see how quickly and how impactful getting this type of sequencing data could be to frontline care. And over the last 3 winters, we've been able to run a metagenomic service in our ICUs for our sickest patients. And if I could say one thing about our pilot studies and the studies that we've been doing, it's not about how we feel. It's about actually what those ICU clinicians actually return to us and say, we want more of this. We need more of this. This needs to be an everyday, every patient procedure. And I think that's the power that we've seen in what we've been able to do with our team here. Most of them are in the room, and Guy had spoke earlier about the previous study. But I think that's the most impactful thing that we have seen with trying to implement sequencing in the front line. And now we want to bring that forward, not only for frontline care, but for also pandemic preparedness and public health. Thank you.
Thank you so much, Rahul. I'm now delighted to welcome our final panel member, Dr. Heather Carlton. Heather is the chief of the Enteric Diseases Laboratory branch in the Division of Foodborne, Waterborne, and Environmental Diseases at the National Center for Emerging Zoonotic Infectious Diseases at the CDC. The mission of the EDLB is to provide quality data, expertise, and effective tools to improve the control and prevention of enteric diseases worldwide. After joining the CDC, Heather led the development of whole genome sequencing-based analysis tools for the transition of the foodborne molecular surveillance network called PulseNet. And she currently serves as the chair of PulseNet USA and co-chair of PulseNet International Steering Committees. Welcome up, Heather.
Yeah, thank you all for having me, and I'll have a slightly different perspective here coming from public health, and give you a little bit of background about what I do, and then I'll have a wonderful assistant walk us through positive disruption if we use approaches like metagenomics and public health surveillance. So I've been at the CDC for 12 years. When I started at the CDC, we were just starting with implementing sequencing for surveillance systems. So I'm responsible for PulseNet, which is our national molecular surveillance system for foodborne, waterborne, and One Health-related outbreaks in the United States. And we also have an arm called PulseNet International, where we work with over 80 countries around the world to help implement this technology for use with outbreak detection within their own countries. Within the United States, 1 in 6 people get a foodborne infection every year. So some of you might have been at my talk and during the innovation days. I know several hands were up for people who've experienced a foodborne infection. And just those averages mean that some of you in this room today will probably get a foodborne infection this year. Sorry about that. But we're trying to improve those odds that we have less foodborne infections. Once you get ill, so that ends up being about 50 million people in the United States that get ill with a foodborne infection every year. If the disease is severe enough, you'll go to your clinician, or you may even be hospitalized depending on the illness and the severity. When you go to your clinician or are hospitalized, and particularly when you're hospitalized, generally a sample is taken then where the identification of the pathogen is completed. And then within United States, there's a requirement that that sample be forwarded to public health if it's one of the enteric bacterial pathogens we track. So we track Shiga toxin producing E. coli, Salmonella, Campylobacter, Listeria, Chronobacter, Vibrio, and a few other bacteria that end up being associated with foodborne outbreaks. We receive about 65,000 samples per year. So remember, 50 million people get a foodborne infection every year in the US. We only receive about 65 isolates within our data system. And even with those lower numbers, we detect between 100 to 400 potential outbreaks each year. When we detect an outbreak, we use signals like the sequence data to find isolates that are closely genetically related, because the assumption here is that they were exposed to the same source. If they're closely genetically related, and then we can go back to those patients and ask them what they ate, and then use that information to kind of triangulate or figure out what the potential common outbreak source was. And because food is a commodity that goes across state lines and is traded globally, having the surveillance system, you have to have a nimble, spread out surveillance system to be able to detect these kind of public health threats. And once we detect an outbreak, we work with our epidemiologists and our regulatory partners at the FDA and at the USDA to help identify that source. So along the path of identifying an outbreak, I think there's several areas where we can use something like Oxford Nanopore technology, and particularly metagenomics implementations to get to the source quicker. And for this example, I've asked Aaron to come up and join me to kind of give a demonstration of what happens now during an outbreak versus where we could go if we implement metagenomic sequencing closer to the point of when you get infected. So Aaron, I'm sorry, but I have given you bloody diarrhea. And now we're going to try to figure out what you have, and I'll walk through an example of our current public health surveillance system and pretend I'm an epidemiologist. I do not play an epidemiologist in real life. I'm a laboratory scientist, to figure out what the source is. And then we'll talk through some examples of maybe when we could have detected the outbreak quicker with metagenomic sequencing. So now you have bloody diarrhea. I mean, if you have bloody diarrhea, most people will go to the hospital to figure out what's the cause. And often for bloody diarrhea, the cause is going to be Shiga toxin producing E. coli. So Shiga toxin producing E. coli, we receive about 6,000 isolates for Shiga toxin producing E. coli each year, generally from your exposure to when you get sick. It's about 2 to 3 days. So common exposures for Shiga toxin producing E. coli are leafy greens, sprouts, red meat, so steak, hamburger, and there's some others too that have been associated with STEC. So can you remember, Aaron, about 2 to 3 days ago, did you have steak, lettuce, or sprouts?
Well, Dr. Carlton, it was yes. It seems like a few days ago, I recall I was at a restaurant, and I did have a bowl of pho. I remember there were some bean sprouts on that.
Okay. Very good. Now let's continue. So now we have identified that he's sick, right? And we've identified, so he's sick right now, 2 to 3 days after he had the bowl of pho, right? But now he's going to go to his doctor and get a clinical diagnostic test. Maybe a culture-independent diagnostic test like the BioFire, which is a multi-analyte panel, and he tests positive a couple days after he's in the hospital with STEC. So now we know it's STEC. So now we're putting you at about 4 days from when you got your food exposure to when you get ill. So do you remember if you ate those things 4 days ago?
It was about 4 days ago. I traveled then, and it might have been, it was at an airport actually that I had that bowl of pho 4 days ago.
All right. So now we know he has STEC, and we tested positive on a culture-independent diagnostic test. It's really important in terms of clinical treatment because antibiotics are actually counterindicated for something like being positive for STEC. And STEC can also cause further complications like hemolytic uremic syndrome. So now we've just gone through the process of we've gotten the clinical diagnostic. We haven't even touched public health yet, right? So now we have a sample that's going from the hospital to public health. It's a sample, so it's not a culture. So that sample takes a couple days to transit, and now it's arrived in public health, and we have to isolate the bacteria. And that process itself can take 2 to 3 weeks. Once we have the bacteria, then we do the sequencing. Our turnaround time is 4 to 7 days in public health. Then that data is transmitted to CDC so we can do our outbreak detection. So if you're keeping track of the amount of time here, that's about 5 weeks, right? 4 to 5 weeks. Okay, so I go back, same question to you. Now I have this information within my PulseNet system. I'm able to detect my potential outbreak, but I'm going back to the case and saying, okay, 4 to 5 weeks ago. So do you remember? Do you remember if you ate red meat? Do you remember if you ate lettuce? Do you remember if you ate sprouts?
Oh gosh, 5 weeks ago, I don't even remember what I ate yesterday.
Exactly. So that's part of the challenge we have in public health, right? We have that diminishing returns from how many people get sick to what actually makes it within public health, what samples make it into public health. And then by the time we've identified a potential outbreak, oftentimes cases when epidemiologists interview them don't remember what they were exposed to. So along that chain, you remember best, I think, what you ate 2 to 3 days ago. 4 days ago, you kind of remembered too. So having opportunities for doing culturing closer to when, or doing metagenomic sequencing closer to when someone gets ill is so critical. So secretly, since we all bring our cell phones into our bathrooms, I would love a small sequencing device. I think ONT has one that you could do your sample collection and sequencing while you're in the bathroom. Your cell phone's already in there. It can help you with the data analysis.
Self-service.
Yeah, there you go. Self-service, public health. But even if people are uncomfortable with that, as we think about where the points were that that Aaron interacted with the clinician and the hospital, there's multiple opportunities to introduce metagenomics and introduce metagenomic sequencing at a level where you could get to strain level subtyping, where we can really improve that time frame so that we can detect the illness faster, understand what's causing the illness, potentially work with clinical and diagnostic companies to share data to understand what's going on sooner so we can get closer, we can detect that outbreak sooner. And by detecting the outbreak sooner, we prevent more illnesses. So I'm sorry I gave you S-Tech, Aaron, but I really appreciate you helping me out with this demonstration.
Anytime, Heather.
Thank you.
Thank you so much, Heather. Great demo. All right, that was fantastic. Thank you so much to all of our speakers. I'd now like to actually invite you all back for our portion of the panel discussion, so we can now start to dive in. So for this panel discussion, we'll be taking questions from the floor as well as the app. And so thank you to everyone. I see we have lots of questions submitted so far, and please keep them coming in, and don't forget you can use the thumbs up icon to upvote your favorite questions. Now with that, this is a really, I think, fascinating panel. We wanted to give enough time in this session to really deep dive. You know, from the perspective, whether they're nonprofits or industry or government, hospital implementation, you know, metagenomics is certainly a common theme across the board, but that still feels a little bit forward-looking, right? And so I think, you know, some of you, including Chuck, you touched on the evolution of different techniques that have been implemented over time. So I'm wondering if each of you could maybe give a little bit more background within your industries. What types of tests have been implemented? How have those tests evolved? And maybe what was that transition period like? And I'm wondering, maybe Rahul, if we can start with you, you know, as someone close to the, you know, the diagnostic tests that are implemented, how have those been changing over time?
Sure. So I think we're one of the first centers to implement a metagenomics test into a pilot service that's now been running for more than 3 years as a pilot. You know, a lot of sites are doing observational work. They're doing some type of interventional work. I think a lot of sites are finding it difficult from a regulatory perspective to start to implement some of these newer techniques, because not only do you have the fact that sequencing is relatively new to the front line, but there are also challenges in sample preparation and the bioinformatics pathways that you need to be able to interpret your results. And putting all of those three new technologies together and then bringing that to the front line can be very challenging. And so I think some sites have started with using isolate sequencing. Others have looked at other areas such as CSF. They've taken a public health perspective. People are trying in blood. But I think in general, it's been very difficult because there hasn't been the confidence to go right into a clinical service. And for us, I think we spent a lot of time trying to perfect what we could, but we felt that it was more important to be able to understand what our limitations were of the test and get that into a clinical service and essentially learn as we iterate rather than wait until a perfect product was available for then to us to do an implementation.
Thanks, Rahul. And Heather, if we can jump to you sort of on that evolution of different tests, PulseNet was started with pulse field gel electrophoresis, correct? And that is not the case today. And I'm wondering, again, if you can give us a little bit of that window into that evolution of the techniques that were used. And again, what was that transition like as you moved towards more genomic approaches?
Yeah, so I started in the enteric disease lab branch in 2012. We started implementing sequencing in 2013 with Illumina. And for PulseNet, we're a distributed network, right? We're talking about state and local public health labs also are part of the network and actually are the ones that are generating the data. So the transition was a bit rocky. We had to figure out a way for all our state and locals to get sequencers. And then we did a lot of work for talking to all sorts of people, epidemiologists, microbiologists, and public health scientists about what's different about the sequence data, how to analyze and interpret the sequence data, and then ultimately validated and developed workflows that are locked down for bioinformatics as well as a well-validated laboratory protocol that led us from starting sequencing in 2013 for our PulseNet pathogens to fully implementing sequencing using Illumina technology in 2019. And we were also one of the first labs at CDC to validate sequencing, where we were the first lab at CDC to validate sequencing for identification of pathogens under CLIA. And we we're the first group to go through the process of doing that validation. And it was interesting. If you guys are familiar with CLIA, those guidelines were developed really for PCR. That was the last time there were updates for it. So as we started to implement this test for identification using whole genome sequencing, having to educate our directors and inspectors about how sequencing is different. No, you don't get the same read every time you sequence the bacterial isolate. Concepts that were obvious to someone working in the field weren't obvious to people that were outside of the field and really used to previous diagnostic technologies. So it was an interesting transition, but ultimately a successful run that's really, revolutionized how we detect outbreaks and, what we're able to use that data for.
Thanks, Heather.
Yeah.
And Chuck, during your introduction, you gave that nice historical context. And again, do you want to paint a little more of a picture from your perspective, maybe on the regulatory side? What is it like when these new technologies are just coming about? What are those pain points or case studies that need to be built when you're really trying to show that clinical utility for new technologies?
Yeah, it's definitely not easy for sure. And from an IVD manufacturer development perspective, we want to understand before we start the process of trying to develop an IVD, does it have value to patient care? If it has no value, we go to all this trouble and difficulty, and it's not easy to develop something that has minimal impact, minimal value to patient care. It's kind of a waste of time. And so one of the things we have to be really certain of from a commercial IVD manufacturer perspective is having confidence we understand what the value is of an IVD so that we develop it and get it through regulatory agencies that it actually will positively impact patient care. And so I think, certainly with new technologies like this, it's evolving, and our understanding of the potential value is not always clear necessarily. And then on the FDA side, they struggle with understanding how to regulate it properly. And so you see a situation like FDA had actually a guidance document for industry for the development of IVDs in the space of infectious disease, and they released that in 2016. There are no FDA approved IVDs using metagenomic sequencing. So they actually withdrew that guidance document in 2023, just last year. And so it's clearly that the case that they're struggling also in terms of understanding what's the best path. What is a guidance document that could create a clear pathway that manufacturers will actually utilize and actually successfully achieve approval with products that will actually improve patient care? And so it really is a challenge that all of us as a community are having to work through. And so and that's a couple thoughts on that topic. So.
Yeah, thanks, Chuck. And Inez, during your introduction too, in that context of pathogen surveillance, obviously COVID-19 had an immense impact on capacity that was built to implement genomic surveillance.
Yeah.
By and large, many of these laboratories had not done sequencing. Maybe they had done PCR or other types of molecular tests, but I think if there's possibly any silver lining for the last few years, it's that more labs have been capacitated to do genomic surveillance and build up the infrastructure. But just do you have other general thoughts on what you've seen in your roles on how labs have gone through that transition into genomic surveillance, and then how that gets maintained going forward? How is that going to be used in this post or peri-COVID era?
That's a really good question, Aaron. And I think one of the challenges we've had is that there was a lot of investment during the pandemic, and then a lot of that stopped as soon as the emergency was over. And so we're trying to find ways to keep that sort of sustainable. And one of the problems we've known from some governments that we're talking with is that a lot of them will have a lot of this equipment, and they've had very specific sort of time restrained training, but then they no longer have the reagents they need to continue to do that sequencing, and they don't necessarily have the training to do it in a lot of the countries that can only do it for COVID. COVID is not their number one priority, as we know, especially now. And a lot of the countries have equipment which is just being mothballed and just sat there not being used. And so trying to make you get the most bang for your buck is what we're really trying to think of. So how can you use equipment for as many different diseases as possible? So in a disease agnostic fashion, where possible. How can you make sure that training is sustainable is really key for us as well. And what is sort of the bare minimum that you need to have available, especially in the resource limited settings that we're focused on, in order to be able to do this well. And like I said earlier, go to those hotspots where we're most likely to see those sort of outbreaks happening. And that is not happening everywhere. Yeah. There are some improvements, and there are other kind of more conversations happening. There's more collaboration. What we do have now is a lot more data. The problem is how to share that data, whether it's being not shared for like political reasons or patient privacy reasons. But that's a very good development, and we just somehow need to work through governance. If anyone has any ideas of how to manage that, I'd love to hear. So we are going in the right direction. We've still got a long way to go. Lots of challenges, I'm afraid.
Yeah, no doubt. Going back to you, Chuck, maybe you had mentioned, some of the struggles that even regulatory agencies have with seeing perhaps the utility. But Rahul, during your, background that you gave us on assessing, these rapid agnostic strategies and the utility that seems very quickly you saw the potential, at least. So I'm wondering, Rahul, if you can, give us your reaction to what Chuck just said about, it seems like there's perhaps a discrepancy there where you're starting to see the potential and yet it's still not clear to everyone yet.
I agree with Chuck that you have to show clinical utility and clinical value. But, for these type of disruptive technologies that, as Chuck had alluded to earlier, that some of the traditional models and ways that we measure clinical utility and clinical value don't actually fit with these type of technologies. And so we need to be disrupting the regulators also, not only disrupting the clinical space. And I think the approach that we've taken is that in order to do that, you actually have to go out and do it. And you can't wait, you have to do it. So you be the first one through the door, get your nose bloody, but make it happen. And I think that's the approach that we've tried to take. And I think, we will be able to inform the regulator with real world information, real world data to show clinical utility. But on the same side, we've learned what the clinical utility is, but we've also learned that the value case might not be there if you look at sequencing alone for frontline care. If you add in the public health perspective, the biosecurity, biopreparedness, pathogen X angles to that, and you find a way to get the proper compensation and monetization of those, then the value proposition changes for your single test. And that's what we're now attempting to do.
Can I ask a question?
Please, yeah.
I just wanted to ask you both a question, all three of you a question. Is there a good example of a regulator who you think is using disruptive approaches and like trying to think about new ways of regulating some of this new technology?
I mean, I think, even if you look at what FDA was proposing before, it was different. I mean, they were suggesting the use of heavy reliance on the Argos database, which they built, which is a reference sequence database with different microbial sequences. And so, they were suggesting an approach that was different. But and when FDA releases these guidance documents, what they're really trying to do is create an avenue for innovation to encourage, commercial companies to develop products that will actually, impact public health and patient care. But it doesn't always work, and sometimes there's a lot of complexity. And sometimes the technology advances fast enough that guidance documents that they create no longer become relevant. And so it's definitely complex and challenging. And I think from the developer's perspective, the biggest difficulty is really understanding with real clear specificity what are the clinical scenarios that we can study and prove value for with an IVD. It's not good enough to say direct from blood in sepsis. That's not good enough. You need more specificity. Are we talking about neutropenic patients? Are we talking about patients with pneumonia? Are we talking about patients with pyelonephritis? And is it an adjunct, or is it something you do afterwards? At what point in the patient workflow do you actually use it? So all of these things are really important to hammer out before you actually go to the trouble of developing an assay and then trying to get regulatory clearance for. And right now, all of that is a little bit confusing. And so I think as more evidence emerges over the next few years and clarifies a lot of those questions, it'll be a lot easier for us to think about how we go about developing an IVD.
I'm wondering, Heather, your thoughts on that from the CDC. You painted a picture for us colorfully of using stool or other samples that could be a monitoring system, but that might be quite broad in scope. Do you think of it too, and sort of piecemeal on how you might give guidance on metagenomics for foodborne pathogen detection?
Yeah, and CDC is not a regulatory agency, but we're definitely impacted in changes in clinical diagnostics and what samples and workflows are now approved. And so right now for our network, we're heavily reliant on isolates, but diagnostics are changing and going to culture independent approaches like the BioFire I mentioned earlier, and there's other multi-pathogen approaches out there. But really, I see this and so you don't have an isolate for public health, and then it delays that whole turnaround time till you get an isolate, then you can do sequencing for. But this is also an opportunity for us to use metagenomics and start thinking about it as a tool for doing isolate-based surveillance, as well as discovering pathogens that haven't been associated with foodborne outbreaks before. About 25% of the outbreaks that are identified within state and local public health labs, the source isn't identified. So the bacterial pathogen is never known. So taking these, a lot of people refer to them as pathogen agnostic. I refer to them as multi-pathogen because you have to know a little bit about what you're looking for to be able to find it. But taking these approaches that are more broad can really impact public health surveillance by detecting new pathogens that you haven't really found in your foodborne surveillance system yet, and being able to disrupt and detect outbreaks sooner. So I think I really watch the clinical space, even though we're not a diagnostic lab per se, because eventually, as clinical labs start generating more and more metagenomics data and do it at a depth that we could possibly get strain level information from, I think there's a great opportunity there for public health to partner with clinical and diagnostic labs to really get information closer to kind of the source of that illness.
Excellent. So we've kind of launched in, but I would love to open it up to the floor for some questions, reactions, please.
I don't know if mine is more of a comment than a question, but it'll lead into a comment. So as a developer, I'm Sean Buckwalter from Mayo Clinic. As a developer, we validate assays. And as you all may or may not know, the FDA came out and launched their new guidance documents for laboratory developed tests, or LDTs. And it's going to be a huge implications for our laboratory. So one comment is this is a great opportunity for Oxford Nanopore and other companies to actually launch FDA approved tests, because that would be a huge relief for some of us developers. But I think about, just some of the comments here about clinical utility, about developing assays and so forth, and just the impacts. I think you were just trying to get into the validation. What do you foresee for some of your metagenomic LDTs and the impacts of this new FDA ruling on your tests and so forth? So I think it's just a great opportunity for companies to do that, and something to think about. It's going to be overwhelming for labs, especially smaller labs. Mayo Clinic's able to do these things, but for the smaller labs that aren't able to validate easily, these companies would have opportunities. So. Thank you.
Chuck, do you have an initial reaction to that?
Well, I completely understand what you're saying, and I think it'll be interesting to see how how we progress through the journey of the attempt to regulate these LDTs in ways that are not disruptive and not disruptive in a way that in any way hampers care to patients. And so I'm not sure how it's going to go, but I think it's an interesting thing that we'll be watching carefully for sure.
We fear the lack of, innovation is not going to be what it would be, because their fear of the LDTs being regulated as they are. And there's a lot to do. It's overwhelming. And if there's little changes in those tests, which we do if we have a laboratory test, and a change happens along the way. So it's just not to, but as a developer, I have to say those things.
Thank you so much. I think we had one right over here, please.
More of a comment really than a question. I found Chuck's presentation especially very interesting, having recently completed an RCT of a bioMérieux product, but I'm now working with Rahul and Jonathan on trying to really bring respiratory metagenomics to the forefront. And I think for any new disruptive diagnostic technology, and the more disruptive it is, the more important it is, I think we really need an interdisciplinary approach. So we're all rightly focusing on the point where we have a product that passes regulators, and that alone is a huge achievement. But then you've got to think about how are people going to use it, and really we need to bring in a really broad community, health economists, but also behavioural scientists, because you can give a frontline clinician a test. They might not do the right thing with it. They might love the test, but it might not produce the actual outcomes and the benefits. I think now's a really good opportunity to do that thinking and do things in a really productive but different way, and bring behavioural psychologists, health economists, patients, and the NIHR here in the UK kind of encourages that approach, and I think it is the right one, but it should be adopted more widely.
Great. Good comments.
Thanks, Vicky. Let's go ahead and take one that was submitted through the app. I think maybe let's think about this. I think it's an interesting concept, metagenomics, as well, because we have surveillance to potential clinical utility, right? And maybe the back and forth and biosecurity, as you highlighted, Rahul. So success of pathogen surveillance relies heavily on international collaboration and data sharing. How can this be incentivized and supported? I suspect this is something you think about, Inez, and do you have any thoughts or comments on how we do create those incentives for collaboration?
Yeah, I mean, like I said, it's tricky. I'd like to answer from the audience.
No one answer.
There's no one answer. I think we need to have governance mechanisms in place. And like I said, obviously the pandemic agreement is being negotiated at the very last hour today. before it's presented to the World Health Assembly next week. I think that should help. We always need to have a plan. I think there needs to be standardized ways of collecting data, which is tricky. And the good thing about resource limited countries where they don't already have lots of the infrastructure in place is you can do things right from the start, collect data in the right format. I think we just need to be as collaborative as possible. You know, we need to stop working in silos. I've heard some horror stories, which I will not share, about people not sharing really important data. Very surprising, always to me. Yeah, I think we just need to encourage that approach. I think there could be something, this might be a bit controversial to say, but I think there could be some commercial incentivizations for all stakeholders involved, you know, and I don't know exactly what that would look like, but as long as along the sort of the chain, all the right people are getting rewarded for it, there's something about having an incentive that will just encourage more use. So I wouldn't shy away from that. It's a scenario I think that we should think about very carefully. And then using all the great technology that we have available today to make sure that a form of the data that countries and citizens are comfortable with is going to be shared and doesn't kind of give people massive concerns about privacy issues.
Well, Heather, I wonder, how do you incentivize users in PulseNet and PulseNet International? It's a very large program. How did that happen?
Yeah, for PulseNet USA, we have a memorandum of understanding of terms of reference. As you join the network, there's an expectation that you share the data, but there's clear guidance on what you share, and if you receive the data from somebody else, what you can do with the data. And I think that's really important, not just domestically, but also internationally, is thinking about not just sharing data is important, but understanding how that data will be used as you share it with others is equally key. And really, what we focus on for our PulseNet International Network, and we're split into 8 regions, is within each region, we focus on building a community of practice. And what's really important for that community of practice is you build trust between public health scientists. So when an emergency comes up, they know someone in that country that they may suspect may be associated with the outbreak, or they may have had cases. Go to that country and they have a trusted network where they can share data between those countries. And that's not something you build overnight, and that's not something you even build in weeks. It takes years to build that network and that community. And that's really been our focus. I think sometimes people can give money for sequencing, and then as a condition of that, you have to upload the data publicly, but it skips kind of the value proposal there of, instead of focusing on having that data in NCBI, you can also focus on having those countries build up how they want to share the data within their region, and then what they need to feel comfortable to share that data publicly. And in some countries too, they just refuse to share data because of previous bad experiences. So a lot of times the challenge doesn't have anything to do with you or your pathogens or anything like that. It's something that happened decades ago. So that building that trust has been really key when you think about that data sharing.
Thank you, Heather. I think we had one right over here. Yes, please.
Over the years, a variety of my colleagues have talked about using aerosol filters at the corner of a room in an airport or in a room like this for surveillance of infectious diseases. First question is, have you seen anything of that sort being functional? And in a room like this, if so, and you had a filter in the corner of the room or a set of filters in the main auditorium, sort of a challenge for Gordon Sanghera, could you detect infectious diseases aerosolized in an environment like this, and sort of prove the case at an Oxford Nanopore meeting?
Anyone want to take a stab at that?
I think you could. So there's different groups that have been doing kind of aerosol monitoring for both biosecurity within regions where you commonly see aerosolized pathogens, like in the American Southwest, for example. There's a few common fungal infections that can be aerosolized, and so having environmental monitoring to understand when those pathogens are in the air and could be causing clinical cases is actually really useful. And some people use that type of monitoring and they take it instead of in a there's a data cave here, right? They'll do air monitoring in a bat cave to understand viruses that are being spread by the bats within the cave, and also understanding what could be emerging in terms of emerging infectious diseases. So I think for aerosols, there's a lot of interesting applications on the metagenomics side. You can understand what's being spread at a conference if you want to. But you can also use it as a monitoring approach too, in terms of biosecurity, to understand what may be emerging or coming. I've seen research projects that do it for a limited period of time, but not routinely.
Were they successful in the sense they detected something in that environment?
Yes.
Thanks, Heather.
Yeah.
Did we have, yes, right down here, please.
Thanks, Aaron, and fantastic panel. I just really want to make a comment if that's fine. Look, we've got public health, we've got a clinical academic centre, we've got industry, we've got a global influencer trying to bring things together. And I think inevitably, as you talk for a while, you end up in seeing the challenges ahead, and it doesn't look like a logical equation. You don't quite know how to fit it together, and sometimes the challenge feels a bit too much. But then you go back and think, well, hang on, what's the unmet need here? And of course, the unmet need is massive. When you turn back to that, you go, yeah, no matter how hard it is, we've got to progress. And of course, people are approaching it in different ways, and they're all valuable. And that's what's great about this sort of room, is that you then hear everybody's different perspectives, and you go away and you mull it over and you make connections, and that will help us bring the innovators to the fore and give them some confidence that it's all right to try something and innovate and fail, and then try something else. And then talk about it, because that might prevent somebody else from making the same mistake. But it'll also then lean down to, well, perhaps the last option standing is the one that you then go for. And I think this is a unique time, actually. We could run the risk as microbiology laboratories of really being left behind in the digital healthcare, AI-enabled genomic future, where infectious diseases is off hospital sites, off academic sites, in a warehouse somewhere that nobody cares about because it hasn't been able to step up to the plate. And that's why I think, the BioFire device has helped us thinking about metagenomics, because people now can see the value of results within hours. I just hope that, as we come back each year, we will see that people in the room now will be the ones talking, and we'll have an increasing number of pieces of evidence brought together until we get to what you were saying, Chuck, which is some kind of study, perhaps not modeled like a drug trial with, like a pharmacological intervention, because it's only a piece of information going through someone's head that has to decide, am I going to just do whatever I want to do anyway? What's the parameters? Some kind of way of gathering that evidence that then can convince clinicians, regulators, developers of IVDs, so that we can take the field forward. So, sorry, a bit of a long comment, but I felt after hearing all these great comments, I just wanted to say that.
Thank you. I really appreciate it, Jonathan. I wonder if I can take a portion of that to bring back to the panel, which is sort of rethinking this framework. Let's go with metagenomics as that theme, where it is a different approach, right? And I think, one thing that we heard from a speaker earlier today, really trying to change the way we think about the diagnostic odyssey, and maybe having an all-in-one form of finding something, but you're not actually looking for anything specific. So how do you change that framework from maybe a regulatory standpoint, or how we provide those studies? Chuck, I wonder if we can start with you on your thoughts on that and how that differs from the way things have been done.
It's definitely challenging, and I really appreciate your comments. I think those are spot on and very appropriate. Yeah, it's interesting. I think as a clinician, I think I'm a little bit sort of torn between the two aspects of it, because on one hand, we don't approach patients in an agnostic way. We have to evaluate them and the information and our exam and the history and whatever lab data we have, and form ideas about what we think might be happening. That's then informed by new technologies and information. And And to think that we could have a situation where we have lab tests that on their own are driving all the thinking is sort of maybe not in touch with the way that medicine is practiced. So I think it is powerful to be able to test for a broader range of microbes and pathogens. And it is very helpful, and it does inform care, but it can't happen in isolation of the evaluation of the patient and all the other information that comes along with the patient. So
Yeah, thanks, Doug. Rahul, what's in your approach? You've been using this routinely
Yeah.
for a bit and starting to expand it to a multi-site. So I wonder if you can continue on that theme, and then maybe with this question that's been submitted, talk a little bit about that transition from your single lab, where you've built the expertise in-house, developed the methods, and now trying to move that to a broader stage of a network within the NHS for the next phase.
Yeah, sure. So, I mean, I agree with Chuck's comments and Jonathan's comments. As a clinician, you develop a hypothesis of what's wrong with your patient. But I think one of the best examples of clinical metagenomics is a patient where you have 3 or 4 clinicians in a room at the bedside, and they've developed a hypothesis that is sound using Occam's razor, the common things being common, and I return them a metagenomics result that's so far in left field that they say, well, we weren't even thinking of that. Okay. And we now need this test because we weren't thinking of that. We would have continued down pathway A until we would have exhausted every possibility. And I think while that's not going to be every patient, there is that bit of the unknown that agnostic technologies are able to provide or inform that does solve some of the questions that we have when we go out to treat patients that we don't know what's actually wrong with them. So I think that's a powerful piece here that we can't overlook. And I don't think that the regulator can't overlook it, but the regulator also has to regulate in an appropriate fashion, which might be a stepwise fashion, similar to how mass spec has been regulated, where organisms are added to a database as they're validated, but you have all the information at hand for you to use if you choose to.
Yeah, thanks, Rahul. Inez, I wonder your thoughts too, in that not looking for everything all the time, but in the context of preventing the next pandemic, you don't know what you're looking for. How do you have a system in place to monitor in those situations?
I mean, that's a good question. I mean, if you don't mind, I wouldn't also mind answering the question that's come for
Please
me.
go for it.
If that's
Dive
okay.
in.
If I can see the screen,
Yeah.
cheat a little bit. But someone asked about how useful is it to train scientists in low resource settings
Yeah,
to perform AMR surveillance if they're in a country where they have limited antibiotics access to them and they have no choice about which antibiotics they can or can't use. And I just want to say, I think it's still incredibly important to collect the data. So much so because the more information you have available to you, the more you can help governments make the best decisions. And I think Shabir Madhi in South Africa recently has seen that a lot of prenatal and febrile deaths are due to bacterial infections with microbial resistance. And previously, if you don't know why kids are dying and you think it's due to malnutrition or other sorts of reasons, you're making all sorts of the wrong policy choices. So I think it's incredibly important. And then if we don't, and I think this will help us get access to better antibiotics and to better drugs in general. So I think, yes, ideally we'd have access, but I do think actually doing some of this more sort of this bigger picture thinking is really important because data helps you make the right decisions, and the type of data you collect is always very political. So if that's...
Please, yeah, that's great, Inez. Thank you.
You're welcome.
Any question from the audience? Any other burning questions that came up? On the note of training, Rahul, back to you, maybe on the network, let's say, of laboratories, or at least the multi-lab site expanding out of GSTT. It's maybe you can give us a little bit more of a picture of what that next phase has looked like so far. How are you training up these labs? What has that experience been like once it moves away from a single lab?
Sure. So I think, we can, having a test run in a single lab essentially is easy, right? You control every aspect of it. You control your space, you control your staff, you control what patients that you're doing it on. And you kind of control the thought process of how a test might be used. The minute you go out to multiple labs, especially multiple labs that already have some expertise in public health or in sequencing themselves, it becomes incredibly challenging to then dictate those protocols, get people to follow along. And then you look at the other side where you're trying to implement these type of protocols in a space that has no experience with sequencing or genomics, and you have a different set of problems. So our experience so far is that there's been plenty of interest and support, but there are issues around how you can implement a single protocol easily in a lab space that's already fixed. I think we'll overcome those challenges, but this is such a disruptive technology that even things like simple IT infrastructure, extraction techniques, space, become a challenge. And but those are some of those challenges that we look forward to trying to solve, because it will make the endpoint product much better when we're able to solve all of them.
Heather, do you foresee similar challenges in metagenomics transition of different sample types, different SOPs? How are you thinking about that moving forward?
Yeah, I think for metagenomics, as was mentioned a little bit before, one of the big concerns we have, in addition to PulseNet, I'm responsible for our National Antimicrobial Resistance Monitoring System, is if you go to metagenomics approach, you can detect antimicrobial resistance genes in that sample, but through most metagenomics approaches, you can't link it back to the particular pathogen. And like you were saying, right, you don't treat a patient in a pathogen agnostic approach. There's guidance based on what is detected in making them ill and what's the best antibiotic to treat them with. So I think there's still a lot of questions to ask there about if you have someone who is CIDT positive for salmonella, like BioFire positive for salmonella, and then you go on to do metagenomic sequencing and detect a bunch of antimicrobial resistance genes, even if it's not in the salmonella, are you going to treat that patient with an antibiotic that their metagenomic sample contains antibiotic resistance genes to? So I think, as we go to more broad approaches like metagenomics, it may or it will disrupt how we think about monitoring antimicrobial resistance, linking it to particular pathogens, and is that really necessary in the future? I think people have a lot of feelings about that, and I don't know that there's good data to treat someone based on antibiotic resistance that was generally detected in their stool sample, for example.
Thanks, Heather. I think that's a great note to end it on, and the pursuit of figuring out how to address infectious disease may also open the door for many, many more questions in that pursuit. So can we have a quick round of applause for our panel? That was excellent.